Hello. Yeah, so this lecture series is going to be an introduction to a couple of lectures that I'll be releasing on eicosanoids. Eicosanoids. So, eicosanoids are 20 carbon, 20, eicosanoids are 20 carbon of organic compounds that are derived from arachidonic acid. So they are derived from arachidonic acid. Arachidonic acid is is also a 20 carbon tetraenoic acid, so which is seen in animal fat. Right? Arachidonic acid is seen in animal fat like meat, like egg, and all of that. Arachidonic acid that can also be synthesized from oil, well, that's from plant materials, primarily synthesized from linoleic acid. Linoleic acid is an octa octadeca dienoic acid. So linoleic acid is an octadecadienoic acid. That means linoleic acid is an 18 carbon fatty acid, while arachidonic acid is 20 carbon fatty acid. So, like I said earlier, I said that eicosanoids are made primarily from arachidonic acid, which can be gotten from diets that are rich in animal fats, or can also be gotten from diets that are rich in plant, plant oil. Or this plant oil, which is rich in linoleic acid, must have undergone some sort of um, elongation and desaturation reaction, which we are going to look at in subsequent lectures. Okay, so now just to reiterate again, say that our arachidonic acid, our arachidonic acid, which is the primary precursor, gives us some sort of a bioactive compound, some sort of bioactive compounds which are called eicosanoids. So these eicosanoids, these derivatives of eicosanoids, we have three primary derivatives that we'll be looking at. We'll be looking at prostanoids, we'll be looking at another derivative I'll be looking at are leukotrienes, which are primarily produced, which are produced by leukocytes, and lipoxenes. So these are the three eicosanoids that are formed from arachidonic acid. So the three eicosanoids formed from arachidonic acid are the prostanoids, the leukotrienes, and the lipoxenes. So what are the importance of these eicosanoids? Like I said, the eicosanoids are formed from 20 carbon uh, polyunsaturated fatty acids, right? So they are very, very much important in that the derivatives of these compounds help to maintain gastric integrity. They help to maintain gastric integrity. And how do they do this? They help to proliferate gastric mucosa cells. So the gastric mucosa cells produce more of mucus. They also um, inhibit or reduce the synthesis of the gastric acid, the stomach acid. Okay, they are also important in renal function. Primarily, they help in relaxation of the renal vessels so as to increase the renal blood flow. So increase the amount of blood that is flowing through the kidney. They are important in smooth muscle contraction also. They are important in contraction of smooth muscles. You can think about their function, um, smooth muscle contraction, like during pregnancy, during parturition, when a woman is about to give birth. Yeah, and also in blood vessel dilation, they help to dilate the blood vessels. They help in platelet homeostasis. Primarily, they are involved in platelet aggregation, yeah, and coagulation. Some of them are involved in coagulation. Some of them prevent platelet aggregation too. So that's hemostasis. Yeah, and they are very much involved in um, inflammatory response. So anytime the body is passing through some kind of stress, induced stress, so the body releases some of these prostanoids, which are which send signals, to the brain to indicate that the body is traumatized. So they are involved in activating inflammatory response and also immune response too. So some of the characteristics of these compounds would be they behave like hormones though, but they are slightly different from the regular endocrine hormones in that they act locally, like their action is local, they are local in action, right? Another function of this is that they are not stored. They are not stored. They are easily metabolized into inactive products. So they are quickly metabolized. They are quickly metabolized into inactive products. Yeah, and they have a short half life. So I think that's understandable because they are quickly metabolized. They are quickly broken down into inactive product. Hence the reason why they've got short half life. So this compound, the first okay, yeah. Before we proceed, we talk about just we look at prostanoids. Prostanoids is what we yes. We said that the derivatives of eicosanoids will be the types of eicosanoids that we get from arachidonic acid are prostanoids, right? Talk about prostanoids. We said leukotrienes and lipoxenes, right? So we'll start by talking about the very first, which are the prostanoids. The prostanoids. So the word prostanoid uh, is from the word. Okay, yeah, prostanoids. No, prostanoids are subdivided into three. Prostanoids are subdivided into three. We've got prostaglandins. We've got um, thromboxanes. So it can be thromboxane A or thromboxane B, but thromboxanes, and we've got okay. So I said and prostacyclin. So these are the three classes of prostanoids that we've got. So we've got prostaglandins, we've got thromboxanes, we've got prostacyclins. So the first that we'll be looking at are the prostaglandins. Now, from the name prostaglandins, poster, to be emphatic on the word poster. So poster, and then glandins. So it comes from the word prostate, like the prostate gland. And why is it named prostaglandin? Why are they named prostaglandins? They are named prostaglandins because they were first discovered in the prostate gland. So they were discovered by a scientist called von Euler. Von Euler isolated these bioactive compounds from the prostate gland. So he believed that these organic compounds are only can only be found in the prostate gland. Sorry, but now we know, in modern times, that the richest source of prostaglandins are seen in the seminal vesicle. So the seminal fluid, sorry, the seminal fluid produce more prostaglandins than any other glands of the body. Now, so we'll be looking at prostaglandins fully. Prostaglandins. And this first lecture aims to talk about the structure of we'll talk about some of the classes of prostaglandins that we've got, like prostaglandin. I'll classify some of these prostaglandins based on the similarity in their functions. So we've got prostaglandins A, prostaglandin B, prostaglandin C, belonging to the first class that I will call class A or class 1 prostaglandin. And so we've got prostaglandin D and E will be classified into class 2 prostaglandin, and we've got prostaglandin F series as class 3 prostaglandin, and I'll talk about prostaglandin G and H. So I'll classify these as class 4 prostaglandins. And also we'll talk about prostaglandin I, which are the prostacyclins. So prostaglandin I, which are the prostacyclins. And lastly, I'll talk about the thromboxanes, thromboxane A and thromboxane B. So we'll talk about the structure of these compounds. So again, the first class we look at is prostaglandins A, B, and C, and the second class we look at is prostaglandins D and E, and the third class will be prostaglandin G, and then the fourth class will be third class is prostaglandin F. So the fourth class will look at prostaglandin G and H, and then we'll look at prostaglandin I, and lastly we'll look at the we'll look at prostacyclin I, prostacyclin, PGI. Okay, so you can see prostaglandin I, which is also called prostacyclin. Prostaglandin I is also referred to as prostacyclin. And then we'll talk about the thromboxanes, like the thromboxane A and the thromboxane B. So now to consider the general structure of prostanoids. So generally, sorry about this. Generally, prostanoids have a cyclopentene ring. All prostanoids have cyclopentene ring with a 7-carbon fatty acid moiety. At the top like this, and an 8 carbon fat, an 8 carbon alkyl moiety below. So this is 1, 2, 3, 4, 5, 6, 7, 8. So this is the general structure of prostanoids. So if we want to number now, we number this carbon to be 1, and this is 2, and then 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. This is 16, this is 17, 18, 19, and this is carbon 20. Right. Now, one thing we must understand is that for prostaglandins, the carbon 15 would always have an hydroxyl group in it, right? So the carbon 15 uh, of the alkyl moiety here has an hydroxyl group in it. All right. So we want to start by looking at the first class of prostaglandins that we created. That's the PGA, PGB, and PGC. Now for PGA, there is a double bond here. Double bond is seen between carbon 10 and carbon 11. So prostaglandin A1. So if I say prostaglandins A1. So this is 1 because it's only having a single unsaturation, which is seen in our cyclopentene ring. And this unsaturation is seen between carbon 10 and carbon 11. So this is prostaglandin A. Now, if I decide to include another double bond, let's say I include a double bond here between carbon 13 and 14. So because there are 2 double bonds now, I call this prostaglandins A2. If I decide to add up one more double bond here, for instance, so now we've got 3 double bonds, so I call this prostaglandins A3. So the subscripts, the number subscripts that we have is dependent on the number of unsaturation in the organic compound, right? So the peculiar thing, what is peculiar to prostaglandin A is that prostaglandin A has an unsaturation between carbons 10 and carbon 11. Okay, that's that for prostaglandin A. Now I'll talk about prostaglandin B. For prostaglandins B, for PGB, our saturation is seen between carbon 8 and carbon 12. So this is our carbon 8 and this is our carbon 12. So the unsaturation is seen between carbons 8 and carbon 12. Why for prostaglandin C? For prostaglandin C, our saturation is seen between carbons 11 and carbons 12. So that's the first class of prostaglandins that we deal with, A, B, and C. We check for prostaglandin A, the unsaturation is seen between 10 and 11. For prostaglandin B, the unsaturation is seen between 8 and 12. And for prostaglandin C, the unsaturation is seen between 11 and 12. All right, so the next class of prostaglandins we'll be looking at is prostaglandins D and E, right? So prostaglandin D and E, they are called beta hydroxy ketones. They are called beta hydroxy ketones. And why are they called beta hydroxy ketones? They are called beta hydroxy ketones because they contain they contain an hydroxyl group at the beta carbon after the carbonyl group. The carbonyl group represents the ketone group now. So one carbon after the carbonyl group contains the hydroxyl group, hence the reason why they are called beta hydroxy ketones, right? So let's look at the structure of prostaglandins D now. Okay, so for prostaglandins D, these are PGD. We have an hydroxyl group on carbon 9. There's an hydroxyl group on carbon 9, and there's a ketone group or carbonyl group on carbons 11, right? So, and there's an unsaturation between carbons 13 and 14. So I'll call this PGD1 because there's one unsaturation. If I decide to include another unsaturation here between carbons 3 and 4, then we have two unsaturation, hence this becomes PGD2. So this is the structure for prostaglandin D. Prostaglandin D will have the beta hydroxyl group on carbon 9 and the ketone group on carbon 11. Now for prostaglandin E will reverse the structure now. Reverse it in the sense that the hydroxyl group, sorry, okay, the hydroxyl group is seen in carbon 11 here. The hydroxyl group here, sorry, is seen in carbon 11, and the ketone group is seen in carbons 9, right? So this will be PGE2 because of the double 2 unsaturations. So this is the structure for prostaglandins E. And now the next we look at is prostaglandin F, which are the diols. So for prostaglandin F series, we talk about the diols. So there's going to be two alcohol groups between carbons 9 and carbons 11. So this would be PGF2 actually, because of the two double bonds. If I decide to erase this double bond here, right, then this becomes PGF, right? So for prostaglandin F, we've got two hydroxyl group between carbons 9 and carbons 11. So that's for prostaglandin F. Yeah. Now we could have geometric isomerism here, in the sense that depending on the orientation of the hydroxyl group in space now, this hydroxyl group could be pointing directly towards us like this. If this hydroxyl group is pointing directly towards us like this, and this hydroxyl group is also pointing towards us, facing the plane of the paper, so this is a kind of cis isomer. So we'll call this okay, let me put introduce the double true unsaturation. So we'll call this PG F true alpha. So we call this alpha to denote cis geometric isomerism. Why? Because the bond carrying the hydroxyl group is actually facing us. It's pointing towards us, right? It's coming towards us. But if the bond carrying one of the bond carrying the hydroxyl group is going away from us towards the plane of the paper, right, and the second bond is coming towards us, then this becomes a trans isomerism. So this becomes like PG F 2 beta, right? So it becomes PGF 2 beta. So beta here denotes trans isomerism. All right, so that's about the PGF series. So we want to look at PGG and PGH. So PGG and PGH are primarily they are intermediates. They are intermediates that are produced in the biosynthesis of every other prostaglandin and prostacyclin, and in fact thromboxanes too. So for these prostanoids to be made, the intermediate, the first intermediate that is generated is the PGG, which is unstable, and PGG undergoes some kind of reduction reaction to become PGH, and PGH serves as intermediate in the manufacture of every other prostaglandin. All right, so PGG and PGH, they are bicyclic prostanoids. So they're bicyclic in the sense that they have been oxidized by an enzyme called cyclooxygenase. So two oxygen has been added to them by the cyclooxygenase in the cyclooxygenase pathway. So they look kind of like this. Right, I'm trying to draw the structure for PGG2 now, and so there's this hydroperoxyl group at the carbon 15, right? So this is PGG2, and this compound is highly unstable. Now, if this unstable PGG2 undergoes reduction, reduction is loss in oxygen, right? So one of the oxygens here becomes lost. So this gives us a more stable PGH2. So this is PGH2. It gives us a more stable PGH2 with an hydroxyl group in the carbon 15, right? So these compounds are endoperoxides, bicyclic endoperoxides. Okay. And lastly, next we'll talk about the prostacyclins. That's the PGI. We can call them prostaglandins I. So for the prostacyclin, if you consider the basic structure of our prostanoid now, what happens in prostacyclin is that there is going to be a ring which is bridging carbon 6 and carbon 9. So between carbon 6 and carbon 9, there is a ring which forms a bridge between these two carbons, 6 and 9. And so this regular prostanoid becomes bicyclic, and so it becomes something like looks like this. You know, we now have a bicyclo a bicyclo pentene ring, right? One of which is carrying a bicyclo here, pentene here. One of which is carrying an oxygen, so the structure kind of looks like this. So the structure comes like this. Yeah. So this is a prostacyclin. So we can see that this oxygen has bridged between carbon 9 and carbon 6. And so we now have a bicyclic compound being formed. These prostacyclins are Primarily, they are like antagonists. They prevent platelet aggregation, unlike thromboxanes, which are very much involved in platelet aggregation. So we'll talk about thromboxanes now. Thromboxanes will be the last class of poster prostaglandins that we'll be looking at. Thromboxanes are also, yeah, they are heterocyclic compounds. Thromboxane A is highly unstable while thromboxane B is stable. They have an oxene ring. An oxene is like a six carbon ring like this, right? And then of course our normal moiety. So this is the oxene ring, right? Our oxene ring. So there is a fusion between this carbon here and this carbon here, and the fusion is formed by an oxygen. So this gives us the structure of this double bond here. This gives us a structure for thromboxane A2, but thromboxane A2 is highly unstable. Now, in thromboxane B2, which is a more stable compound, this is formed by the bond dissociation of this bond and this bond, the break. I guess it's some kind of an hydrolysis reaction. And what happens eventually is that a diol is formed, a diol is seen on the oxane ring. This is one hydroxyl group, and this is another hydroxyl group to form a diol on the oxane ring. So this is a more stable compound, which is our thromboxane B2. So thromboxane B2 is more stable compared to thromboxane A2. So that is all for the structure of prostaglandins. So quickly, we'll look at some of these prostaglandins and where they are seen. Yeah, I mean the occurrence of these prostaglandins. So first we talk about the seminal fluid. So like I said, the seminal fluid contains the highest proportion of prostaglandins that we find. So inside of the seminal fluid we have prostaglandins E1, find prostaglandins E2, we find prostaglandin F1 alpha, we find prostaglandin F2 alpha, we find hydroxyprostaglandin E1, hydroxyprostaglandin E1, specifically 19 hydroxyprostaglandin E1. We find 19 hydroxyprostaglandin E2. We find 19 hydroxyprostaglandin F1 alpha, and we find 19 hydroxyprostaglandin F2 alpha. So these are the numerous numbers of prostaglandins that we find in the seminal fluid. There are about 1, 2, 3, 4, 5, 6, 7, 8, about 8 prostaglandins. Yeah. Or much more I'll find in the seminal fluid, and they all have the functions for integrity of the sperm, relaxation of the smooth muscles of the vaginal and the uterine wall, and all of that. So, okay. So next we look at the menstrual fluid. Within the menstrual fluid, or the menstruate, if you will, the menstruate has two specific types of prostaglandin. We have the prostaglandin E2 and the prostaglandin F2 alpha. So these are the two types of prostaglandins that we'll find in the menstruate. Also the amniotic fluid and the endometrium. That's during labor, the amniotic fluid and the endometrium produces prostaglandins E1, prostaglandins E2, prostaglandins F1 alpha, and prostaglandin F2 alpha. So these are seen in the amniotic fluid and the endometrium during labor. Right. So also we'll talk about the maternal blood during gestation and labor. So the kind of prostaglandins that are produced by the maternal blood during gestation and labor will be, there are also numerous too. So yeah, okay, we've got prostaglandin F2 alpha and prostaglandin E2 alpha, which are produced by the maternal blood during gestation and labor. And also we'll talk about the prostaglandins that are produced by the fallopian tube and the decidua of the uterus. So the decidua of the uterus will produce prostaglandin E2 and prostaglandin F2 alpha. And lastly, the umbilicus, the umbilical cord. So what kind of prostaglandin do we find in the umbilical cord? So in the umbilical cord, we find prostaglandin F2 alpha. Just to revise, we started by talking about arachidonic acid and how arachidonic acid helps to form these prostaglandins, right? So, and then I chipped in by saying that the primary intermediates that are involved in the formation of most prostaglandins are prostaglandin H. Prostaglandin H is the more stable form of prostaglandin G, right? So prostaglandin G is unstable, and the more stable form is prostaglandin H, right? So now prostaglandin G1, okay, sorry, prostaglandin G. So we said the more stable form is H. So starting from prostaglandin E, now I just want to chip in one or two things for my you guys here. So prostaglandin E1, that's the prostaglandin with one on saturation, are derivatives of Eicosanoids 8, 11, and 14 trienoic acid. And I think that's the same as homogamma linoleic acid. So prostaglandin E1 is a derivative of gamma homolinoleic acid, or 8, 11, and 14 trienoic acid. Again, I said gamma homolinoleic acid, right? So for prostaglandin E2, are derivatives of 5, Eicosa 5, 8, 11, and 14 tetraenoic acid. No, it's tetra because we have 4 double bonds now. Double bond on 5, double bond on 8, double bond on 11 and 14. So 5, 8, 11, 14 tetraenoic acid. So specifically the name will be Eicosa 5, 8, 11, 14 tetraenoic acid, which is ultimately the same thing as arachidonic acid. So the precursor for prostaglandin E2 is arachidonic acid. The precursor for prostaglandin E1 is gamma homolinoleic acid, and the precursor for prostaglandin E3 is Eicosa 5, 8, 11, 14, and 17. So we've got 5 double bonds now. Eicosa 5, 8, 11, 14, 17 pentanoic acid. I'll take it again. So for prostaglandin E1, so it's derived from gamma homolinoleic acid or Eicosanoids 8 11 14 trinoic acid. Prostaglandin E2 is derived from arachidonic acid, which is 5 8 11 14 tetrahinoic acid, or specifically we say Eicosa 5 8 11 14 tetrahinoic acid. And prostaglandin E3 is derived from Eicosa 5 8 11 14 17 pentanoic acid. So in the next lecture, we'll be talking about the pathway to the biosynthesis of prostaglandins, right? So we're talking about the cyclooxygenase pathway, how these prostaglandins are formed from eicosanoids, from arachidonic acid. Okay, before we talk about that, first I'm going to describe how arachidonic acid is incorporated into the membrane phospholipid, and after the incorporation of arachidonic acid into membrane phospholipid, how arachidonic acid is being cleaved from membrane phospholipid and then released into the cell and utilized by the cell in a specific pathway called the cyclooxygenase pathway, so as to manufacture or make prostaglandins. So I'll see you in the next lecture.